Christina Annunziata got her MD, PhD at Georgetown University Medical School, and she did a residency in internal medicine. She came to NCI and trained with Blue Stout. Subsequently, she was in the Clinical Investigator Development Program as an assistant clinical investigator, and now she's in the Women's Malignancy Branch. She directs clinical operations, and she's head of the Translational Genomics section. And her title today, Ovarian Cancer in the Genomics Era. Dena.
Great. All right, thank you. Let me do this. Okay. All right, and then this one is on too, right? So I can look in both directions. Okay, so today I'll talk to you about ovarian cancer. And so this lecture is going to be, the beginning will be about ovarian cancer in general, and then I'll go into what we have known about some of the molecular aspects of ovarian cancer, and then I'll finish with some of the research that we're doing right now. What is genomics? So genomics is, in my opinion, the study of the whole genome. So this is chromosomes, gene expression, and global analysis of molecular aberrations of genome of genetics, and not just individual entities of genomics of genes. So we can think of the genomics era actually as starting back in 1959 when Noel and Hungerford studied the chromosomes to identify a recurrent genetic abnormality in patients with a type of chronic leukemia. Since they were in Philadelphia, they called this genomic abnormality, they called this the Philadelphia chromosome. So what is the Philadelphia chromosome? They actually found this little tiny thing. If you can see here, this is an abnormal snippet of a chromosome here, marked by the arrow. And they found that this was recurrent, and this occurred in pretty much everybody who had chronic myelogenous leukemia. So they were looking at the whole chromosomes to find this one aberration. Then it wasn't until 1973 that Janet Rowley found that this chromosome was probably an aberration between 9q and 22q, because those parts seem to have been flipped in the karyotype of the patients. Then it took till 1984 that Graffin et al. Discovered that this was actually a fusion. There was a fusion between the breakpoint cluster region, which is called BCR, and the kinase ABL, which is an ABL kinase from chromosome 22. And then it took until 1996, where Dr. Drucker found a compound that actually blocks the kinase activity of ABL kinase. And then this drug eventually became approved for the treatment of CML with this particular translocation. So that was kind of a long process. So how can we sort of speed this up? So for this, we could actually look at what I call functional genomics, looking for what part of the genome is actually functional, what is causing an effect, what actually transforms normal cells into cancer cells. And here we're looking for what people are calling driver alterations. So an example of this is that in 1981, She et al. Discovered HER2/neu. And the way they found this was actually they looked for genes from, or pieces of chromosomes actually, from carcinomas and neuroblastomas that would transform mouse fibroblasts that were not otherwise cancer. So they kind of like shotgunned in the whole pieces of chromosomes from these cancers into normal fibroblasts. And in 1984, they discovered that this NEU, so it's called NEU from neuroblastoma, had a lot of similarities with EGFR, which is epidermal growth factor receptor. And this is a southern blot back in 1984. And then 1985, Cousins discovered that this was actually HER2, so she called it HER2, which is human epidermal growth factor receptor 2, and that seems to be amplified on chromosome 17 in patients. And in 1987, Dennis Slayman et al. Discovered that this HER2 was amplified in the genomes of several breast cancer patients. And then from here, I don't have a slide, but from here, a drug, an antibody was developed targeting HER2 that is called Herceptin and is now used for treatment of women with breast cancer that have this particular aberration. So that is truly a targeted therapy gotten from a functional genomics approach. So what about ovarian cancer? Do we have any drivers in ovarian cancer? So just some quick background on ovarian cancer. Ovarian cancer is the most lethal gynecologic cancer in the US, with greater than 16,000 women dying per year of ovarian cancer. It's the 5th most common cancer death in women. And this is because 70% of these cases are diagnosed with advanced stage. There's no early test, there's no early detection of ovarian cancer. Less than 35% of patients with advanced stage disease are alive at 5 years. And here you can see the breakdown. So stage 1 ovarian cancer is confined to the ovaries. That's found in about 20%, only about 20% of the time, but fortunately the survival when it's confined to the ovaries is 90%. When it is confined to the pelvis in stage 2, this is a very uncommon place to diagnose ovarian cancer, but there's still a good overall survival of 65%. Most of our cases are, so 75% of the cases are discovered in, diagnosed in stage 3 and stage 4, where you can see here the survival is much lower. So what's the treatment? What do we do for women with newly diagnosed ovarian cancer? First, we do complete surgical staging, optimal reductive surgery, chemotherapy, or clinical trials. So chemotherapy is typically a platinum, either cisplatin or carboplatin, in combination with a taxane, typically paclitaxel, but sometimes docetaxel can also be used. If the patient is diagnosed with stage 3 and has had an optimal reductive surgery, meaning that nearly everything was taken out at the time of surgery, then she can get the therapies intraperitoneal, and that means injecting the chemotherapy directly into the abdomen, into the peritoneal cavity. And that has been shown to have a much better overall survival, actually an increased survival of women by a year and a half on average. And you could see here, this is a quick timeline of the advances in ovarian cancer chemotherapy. In the 1960s, we only had alkylators, and that's such, for example, melphalan would be a type of alkylator therapy. In the 1960s, when we only had these drugs, no women lived beyond 5 years if they had stage 3 or stage 4 ovarian cancer. In the 1970s, with the introduction of cisplatin, 5% of the women were now alive at 5 years if they had advanced stage disease. Then combination therapies became used in the 1980s, and we got even better survival, up to 15%. With the introduction of the taxanes was a big bump. So in the 1990s, there was the introduction of paclitaxel, and here now we're having women, 35% of women with advanced stage disease are living 5 years or more. And then in the 2000s, we have the same 2 drugs, but we now give them in the intraperitoneal location, specifically in women with stage 3 ovarian cancer that have been completely resected. And now we can get the survival up another 5%, up to 40%, just by learning how to use the drugs in the most optimal fashion. But what is ovarian cancer? So ovarian cancer is not one disease. So you can see here, based on these just histologic images, there are various different histologies or various different types of ovarian cancer that we kind of group all in the same category for treatment purposes. So serous ovarian cancers are the most common. Then we have endometrioid ovarian cancers. We have a type called clear cell, and you can see here these are probably, I don't know if it's too small to see, but you can see clear cell, there's cells that look clear, so we call that clear cell ovarian cancer. Mucinous ovarian cancer that secrete a lot of mucin. sorry, that's up here. And then other meaning non-differentiated, where we can't really tell what it is because it's kind of too undifferentiated to tell. So we have all these things that look very differently under the microscope, so they probably come from different places. And in fact, more recent studies have shown at the genetic level, probably serous cancers maybe come from fallopian tube and not from the ovary itself. So this is a type of epithelium. It has a type of signature in mutations that seem to show up first in the fallopian tubes, especially in women with hereditary ovarian cancers that have had their fallopian tubes removed for prophylactic purposes. Endometrioid and clear cell cancers maybe come from the endometrium, which maybe, so in women who have endometriosis, which is a type of syndrome where they have retrograde menstruation, so the endometrium actually ends up in the abdominal cavity. These women potentially get endometrioid and clear cell histologies of ovarian cancer. Mucinous cancers, it's not clear if these come from the gynecologic tract or if these are just metastases from appendiceal cancer, for example, or some other GI source. So there's a lot of different theories now that maybe these are coming from different places, maybe they have different molecular aberrations, and maybe we should treat them differently. So with ovarian cancer, we hypothesized that increasing our understanding about the biological and biochemical events underlying progression of ovarian cancer will create new avenues for treatments. And we asked, can we use genomics to figure this out? So in clear cell and endometrioid cancers, there was okay, just as a reminder, these are only 5 to 10% of all cases, whereas serous cancers are 70%, which is the most common. But in these 5 to 10% of clear cell ovarian cancers, they have a worse outcome with respect to responses to standard chemotherapy and overall survival. They tend to be associated with endometriosis. As I mentioned, maybe in up to 40% of cases, there has been a preceding diagnosis of endometriosis. So back in 2010, there was a group in Canada that used genomics to study clear cell ovarian cancer. And what they did was they started out by sequencing RNA from 18 clear cell ovarian cancers and 1 cell line in their discovery phase. And then when they found aberrations, they actually went back to another 210 samples and sequenced the DNA exons from 101 more clear cell. And here now they also included the endometrioid. They looked at serous as a control and 1 more cell line. And then they went back and they looked at the protein in another 455 samples. And this is very busy, but the point of this is to show you that there was a gene, 1 gene locus was found to be recurrently mutated in cases of clear cell ovarian cancer. And the mutations were all across the gene locus. So this is ARID1A, and it can be mutated in either clear cell or endometrioid cancer. So what is ARID1A? Well, ARID1A is a member of the SWI/SNF chromatin remodeling complex. So it's a DNA, basically a DNA repair or DNA remodeling process. It's mutated, it has been found previously to be mutated in a few % of breast cancer and lung cancer. And across all cancers, this gene, this locus of 1p36 has been shown to be deleted in 6% of all cancers. So when something is deleted, we think of it as not an activating mutation, but probably loss of a tumor suppressor. So possibly this is a tumor suppressor gene. At the protein level, the protein is called BAF250A, and you can see here that with patients who had the ARID1A mutation, 73% did have loss, this is loss in the y-axis, did have loss of staining for the protein. This is clear cell clear cell ovarian cancer. This is endometrioid ovarian cancer. And you can see here in none of the high-grade serous cancers, what did the mutation occur. So ARID1A was mutated or lost in over 40% of clear cell, 30% of endometrioid, but less than 1% of serous cancers. So it does seem to be pretty specific to clear cell and endometrioid cancers. However, it has an unknown oncogenic mechanism. Therefore, we don't have a clear therapeutic utility of this. And so maybe this is a diagnostic utility, but this experiment, if you keep in mind the introduction of this talk, this was not a functional experiment. This was kind of like a big overview, shotgun looking at things. So they did not use a functional approach to identify this. So we're left with maybe a diagnostic marker at this point, but unclear what we're going to do with it from a therapeutic standpoint. Mucinous cancers, I don't have much to say about mucinous cancers. They are very rare, and it's not clear that they are actually from ovarian origin. I just have a couple of slides on mucinous cancer just to show you that, in fact, mucinous cancers here in the bottom have a worse overall survival compared to other types of ovarian cancers when treated with standard chemotherapy. There was a study done here actually at the NCI by Mike Buer when he was an investigator here. And he showed that from the genomic level, so he did gene expression profiling of serous cancer, of normal ovarian serous epithelium, that's what OSE stands for, low-grade serous cancers, high-grade serous cancers, and mucinous cancers. And he found that from a gene expression signature standpoint, the mucinous cancers all grouped together, whether they were high-grade, low-grade, or whether they were high or low-grade. So the mucinous cancers appear to be a different entity based on global gene expression profiling. From this, actually, in the cooperative group, we went ahead and actually made a separate category for mucinous cancers. So now when we're designing large clinical trials, mucinous cancers have their own set of trials. They're not included with the high-grade serous because of this. So this is actually an important finding. One of the things that came out of this was KRAS, but this was a very small study, and this is still sort of in the hypothesis stage. It's not clear whether KRAS could be a target in mucinous cancers. So what about low-grade serous cancers? So we actually have a little bit more clinical information on low-grade cancers. So we are now considering low-grade serous and high-grade serous to actually be two separate entities. And this is because of some of the molecular findings. So low-grade serous cancers have frequent mutations of KRAS and BRAF. In a study looking at low-grade serous compared to borderline cancers, which are a type of low-grade, compared to high-grade serous cancers, there was KRAS and BRAF mutations found in greater than 60% of these low-grade cancers and no cases of high-grade cancer. So that kind of gives you an idea that it's not just like a continuum from low-grade to high-grade. They're actually two separate entities. And this is just a graphical representation showing you that the low-grade cancers have KRAS and BRAF mutations. High-grade cancers do not. Endometrioid and clear cell rarely have it, or less commonly, I should say. So what about KRAS and BRAF? So the RAS-RAF pathway signals downstream of growth factor receptors to, so RAS can be activated and can signal through RAF, MEK, and ERK. So there was a thought that perhaps we could use MEK inhibitors in low-grade serous cancers. So downstream of RAF, we were blocking at, of RAS and RAF, we were blocking at the level of MEK. There was a study that was published in 2013 for women with low-grade serous ovarian cancer treated with a MEK inhibitor. They treated 52 patients. 8 patients had a response and 34 patients had stable disease greater than 4 months. So you would think, let's look at the KRAS mutations, and maybe the ones with the KRAS mutations were the ones that responded, right? Well, no. So we did look at these people did, they looked at KRAS mutation status, and actually, here I just circled right down here, patients who had the tumor response, most of them did not have a KRAS mutation, and 2 of them did, but also 14 patients who had a KRAS mutation did not respond to the MEK inhibitor. So this was very disappointing because we thought we had a molecular target and a specifically defined subset of ovarian cancer, but this did not work. So back to the drawing board with this, and actually we have a couple more avenues for low-grade serous cancers that are being studied right now. And the reason why it didn't work is because probably we think of RAS signaling through RAF and MEK, but in fact, RAS signals through lots of other stuff and not just RAF and MEK. So probably we blocked here and it could go crazy all over here and still cause the cell to grow. So we either need to block up here or we need to block other pathways. And like I said, there are studies underway looking at a lot of other options. Okay, so what about high-grade serous cancer? So this is what we study mostly here at the NIH, high-grade serous ovarian cancer. So there was a genomic study back in 2011. The Cancer Genome Atlas study for ovarian cancer was first published. Cancer Genome Atlas has clinically annotated high-grade serous ovarian cancer samples, and the goal was to identify molecular abnormalities that could influence pathophysiology, affect outcome, and constitute therapeutic targets. So what they did was a microarray analysis of 489 cancers. They looked at messenger RNA, they looked at microRNA, they looked at DNA copy number, and they looked at promoter methylation. And in 316 of those, they looked at whole exome sequencing. Sample inclusion criteria, these were all newly diagnosed women, so they were not relapsed ovarian cancers. They all had serous adenocarcinoma. They had no prior treatment, and they had to have a companion normal tissue specimen. This normal tissue specimen did not have to be ovarian tissue. So they had have adjacent normal tissue, but this was only in like less than 10% of women had these. Otherwise, they got germline DNA from blood cells. So what you can see here is that there were copy number abnormalities all over ovarian cancer. So what this is showing you here on the y-axis is all the chromosomes, 1 through 22. Across the x-axis, so to speak, is individual cases. So each column is an individual specimen of ovarian cancer. And here is it comparing to glioblastoma, which was actually the first tumor that was went through the Cancer Genome Atlas. So what you can see here is red is up, red is amplification, and blue is deletion. So you see here in glioblastoma, you have very nice one area that's recurrently amplified and one area that's recurrently deleted. But otherwise, it looks pretty quiet across the whole genome. But you see here in ovarian cancer, there's like craziness. So it's just genomic disarray across the whole genome. You do have recurrent areas of amplification and deletion, but there's like too many to focus on. So there's a lot of genomic disarray. So why might that be? Actually, one of the recurrent themes here in ovarian, well, the P53 was the most common mutation, and that seemed to occur in pretty much all cases of high-grade serous ovarian cancer, either mutation or deletion. But you also see here that these genes, BRCA1 and BRCA2, they were sort of the next most common, because if you put them together, 11, you had 22%. So they were the next, 22 cases with mutations, the next most common pathway to be mutated. And these are involved in DNA repair. So that kind of gives you a hint that maybe there's this whole theme of DNA repair defects in ovarian cancer that's causing these genomic disarray. So here you can see that this was looking at not only amplifications, but they were also looking at methylation here in the green. So these were the mutations, not amplifications. These are the mutations. The green is the methylation, and then here is just downregulated gene expression. So 33% of the cases appeared to have some alteration in BRCA pathway. And when they looked at the outcome, actually the patients with the mutated BRCA genes had a better overall survival, better response to standard chemotherapy than women with either the methylation or the wild type. And what we're talking about here is that the homologous recombination, that's what HR stands for, which is one of the pathways of repairing double-strand DNA breaks. Homologous recombination pathway was altered in almost half, or a little bit more than half of cases. So this could potentially explain, like I said, the DNA disarray. So you can see they found mutations in ATM, in ATR, BRCA, MC, RAD51, PTEN. These are all, FINK52, these are all in the complex of double-strand DNA repair. And this is just summarizing it again. This pathway is potentially important in about half of cases, ATM, CHEK2, BRCA, in the DNA repair pathway. And then the other half of cases appeared to have problems with cell cycle progression. So what next? Are there new therapeutic approaches? Well, in the 50% with the homologous recombination defects, perhaps we can use drugs called PARP inhibitors, and I'm going to talk about that in the next couple of slides. We can look at commonly deregulated pathways, such as the FOXM1 and Notch signaling, that might provide opportunities for therapeutic treatment. And what they noted in the Cancer Genome Atlas publication is that they've had inhibitors in the clinic already that existed for 22 genes in these regions of recurrent amplification. So now the theme is not looking for one particular gene that's a driver, but maybe we're looking at genes or networks or pathways that can be targeted based on their global effect. So what about PARP inhibitors? So PARP inhibitors are one of the things that we've studied in our clinic, targeting the homologous recombination deficits that could be potentially relevant for half of women with high-grade serous ovarian cancer. So the homologous recombination pathway has not been new. Homologous recombination BRCA mutations were found in 1990 by Mary Claire King et al. And she published this pedigree in breast cancer patients where there was a family history of breast cancer, and she located this genomic locus that contained BRCA1. In high-grade serous cancers, we have not only BRCA1, which only accounts for 8% of ovarian cancers, we also have BRCA2. We have somatic mutations, so these are just in the germline, accounting for 14%. It can also be mutated only in the tumor itself and not necessarily the genome of the patient. Both BRCA1 and BRCA2 account for that. BRCA1 methylation, although that's questionable with respect to functionality. The MC amplification, PTEN loss, and other homologous recombination defects account for almost half of ovarian cancer patients. Beyond BRCA mutations, looking for other signatures of patients who might have a functional homologous recombination deficiency. This group in 2014 came up with a gene expression signature, and they found that actually the patients with gene expression signature had a better overall survival with respect to standard chemotherapy compared to the homologous recombination intact. And so this is actually quite similar to looking at BRCA itself. And you can see here, even in cell lines, the response to a PARP inhibitor was much better if you had a deficient BRCA pathway, or a homologous recombination pathway, compared to if you had intact homologous recombination. So what is a PARP inhibitor? So PARP inhibitors, like I said, we're targeting them to BRCA mutant cancers and other women with homologous recombination defects in that pathway. So as you know, with cellular metabolism or other environmental exposures, you get typical single-strand DNA breaks. So this can happen sort of just, with normal environmental exposures and not necessarily with any other specific insult. So you can get a single-strand DNA break. And PARP comes in, and the PARP labels these single-strand DNA breaks so that the single-strand DNA breaks can be repaired by one of the pathways that is involved is base excision repair. But anyway, the PARP enzyme labels this so that it can be repaired. If you inhibit the PARP enzyme, then when cells replicate, they have a natural process, as you know, of nicking the DNA so it can unwind. And that nick, if it occurs near a single strand break, will then effectively cause a double strand break. When a cell is normal, it has this mechanism of homologous recombination that it can repair this homologous recombination, it can repair this double strand break and continue to live. However, if a cell does not have BRCA, or if it has some defect in the homologous recombination pathway, there's not effective double-strand DNA break repair. You don't have homologous recombination, and you actually get cell death. So because these double-strand breaks overwhelm the cells that don't have homologous recombination, they are exquisitely sensitive to PARP inhibition. So olaparib was one of the first orally available PARP inhibitors. It's now approved for use in ovarian cancer with BRCA mutation. It was one of the first orally available PARP inhibitors, and it was shown to be synthetically lethal. Here you can see in cell lines which had BRCA intact. They were pretty resistant to the PARP inhibitor. With BRCA2 knocked out, they were intermediately sensitive. And with BRCA1 knocked out, they were exquisitely sensitive. So we did a phase 1 and 1B study of olaparib, which is the PARP inhibitor, in combination with carboplatin. The purpose of the carboplatin was to speed up these single-strand DNA breaks. So carboplatin is a chemotherapy that will cause damage to the DNA in single strand, and then the single strands will break. So we wanted to speed that up and not just wait for environmental exposure, speed it up with the carboplatin, and then prevent the homologous recombination, prevent the single strand repair with olaparib, so we would get double strand breaks. So we had 3 cohorts. We had one cohort with women who had BRCA mutation. We had another cohort with triple negative breast cancer. They had normal BRCA, but we hypothesized that they would have similar molecular aberrations. And then we had another cohort with high-grade serous ovarian cancer, which we suspected 50% of these would potentially be sensitive to the PARP inhibitor. So we actually escalated the PARP inhibitor and our carboplatin until we could find safe doses of each, and we're able to give them safely together. And all of these have been completed so far, and we have published back in 2014 the results of the BRCA mutation cohort. So we enrolled 45 patients. 37 of them had ovarian cancer and 8 had breast cancer. With this was a phase 1 dose escalation where we had 30 patients to find a safe dose. And then we added another 15 patients in an expansion cohort at the maximum tolerated dose, which was carboplatin of AUC5 and olaparib 400 milligrams twice daily. So we found remarkable activity. So this is actually very dramatic. So what you have here on the y-axis is the percent that the tumor shrunk. If we see a shrinkage of 30% or more, we consider that to be a true response. If we see growth of 20% or more, we consider that to be progression. And anything between 20% high and 30% down is considered stable disease, just because of the variability in the measurements on the CAT scan. But you can see here that more than half of the patients had a response. So this is actually very dramatic. I mean, these patients had multiple previous chemotherapy regimens that they were resistant to, and the tumor just kept coming back. And here we can see that we're getting a response in more than half of them. So this is very, very exciting. The little stars mean that the patients were still on study at the time of the publication. And then these colors mean the low dose all the way up to the high dose at 5, and then dose level 6 was the expansion. So we have most patients in the expansion. Okay, so that's very exciting. This is a summary just showing you that we had no patients who had a complete response. We had 15 patients, 44% had a partial response, 38% had stable disease greater than 4 months, and 6 patients, or 18% had progression as their best overall response. So the duration is actually also important. So we still actually have patients on this study, and they've been out treated for more than 45. So patients are out more than 4 years. So this is really exciting. So the conclusions was that oral olaparib, which is the PARP inhibitor, is well tolerated in combination with carboplatin, highly active in advanced chemotherapy refractory BRCA deficient cancer, greater activity, of course, at the higher dose, and proof of concept that the activity and tolerability of genetically defined targeted therapy in BRCA deficient cancers. So very exciting stuff for high-grade serous BRCA mutation. But what about new targets? So now we're trying to look at what can we use from a functional standpoint. So the Cancer Genome Atlas, even though it was very extensive, Cancer Genome Atlas was not a functional approach. It was just a global survey. So how can we use something functional to figure out what is actually important to these cancers? And this is now looking for what we are potentially using as actionable mutations, although this is a very vague definition. There are various tests out there, either from Caris or Foundation One, that report possible or unlikely benefit based on the literature. There are clinical trials going on to put these mutations in baskets, but in these trials, there's actually no functional hands-on link. These are all just sort of, we see it there, but we're not really sure what it's doing. So this is actually a very interesting article if you want to read it about the definition of actionable mutations. And these people are very insightful, saying that to call it actionable depends in large part on the strength of the data linking the target to the targeted therapy. And for this trial design to work, 2 key conditions must be met. Number 1, the tumor has to depend on the target pathway. And number 2, the targeted therapy must reliably inhibit the target. So this is like no-brainer for if you work in the lab and you know, of course, this needs to be true. But when you're trying to take this to patients in large scale, this is a very tricky area. Achieving both goals can be a matter of some complexity. So how can we find what is truly, truly actionable, that the cell actually depends on? So I think, in my opinion, we need a functional experiment. And for this, I've used functional genomics. And this is just a very brief overview of one of the things that we're doing in my lab. Back in 2006, my former postdoc mentor actually published that you can use functional genomics to identify targets. And for this, he created an shRNA library where he has shRNAs targeting specific genes and barcodes to identify whether the construct has been incorporated into the cell. At this point, they were targeting 2,500 genes, but now they're up to about 5,000. 3 shRNAs per target. Now he's up to 6 to 12 shRNAs per target. And the library targets basically functional kinases, deubiquitinating enzymes, and NF-kappa B pathway. So what does this mean, an shRNA library? I'm sure you guys are all familiar with this, but just to briefly go over it. The shRNA library is a shRNA construct with a barcode. These are infected into a pool. The pool of shRNAs is infected into the cancer cells. And then you can turn on the expression in an inducible fashion, or you can leave it off. And these are turned on with the TET repressor system. So the TET repressor is always there. So in the off system, we don't give it doxycycline, and in the on is we do. So, and then what we look for at the end of a 21-day growth is shRNAs or barcodes that have dropped out of the population where the shRNA was on, and not in the ones that had the shRNA off. So we're looking for things that are actually blocking cell proliferation or cell survival. We PCR amplify the barcodes, and actually we used to do a barcode microarray, but now we just do barcode sequencing. So we did this in 4 ovarian cancer cell lines. We did 2 serous cell lines and 2 non-serous cell lines to look for functional targets. And what we found was that actually we could find 63 shRNAs representing 55 genes that were common to at least 3 of the cancers. We followed up on these, and actually some of the interesting ones we put into another mini library for validation in more cell lines of the serous ovarian cancer type. So what we have done is that we followed up in 6 additional cell lines using 2 different RNAi constructs for each of these 55 genes, and we actually focused more on the ones that have drugs in the clinic that we can then use. We focused the functional screens on the specific subgroup of serous ovarian cancer, and we did combination matrices to look for what's going on there. One of the most interesting genes that we found is CHEK1. We found that CHEK1, actually, when we went, this was a highly significant in our shRNA library and in the validation screen in the 6 serous ovarian cancer cell lines. When we went back to the Cancer Genome Atlas, we found that it was actually overexpressed in nearly all of the high-grade serous cancers compared to the non-cancer tissue. So what we did in order to hone down on this, we actually went to all the shRNAs and then looked for the 65 genes, as I mentioned, and then came down to 3 genes that were the most significant and the most highly overexpressed in ovarian cancer in the Cancer Genome Atlas. So what is CHEK? So CHEK is actually in the cell cycle pathway, but is also involved in DNA damage repair pathway. So when DNA is damaged, it activates the DNA damage response, which is shown here by DDR. And that signals to activate ATM, which activates CHEK1 and CHEK2, that actually turns off the CDC25. So it turns off the cell cycle. The reason why it turns it off is so that the cell can have time to then repair the DNA before it goes through the cell cycle and completes the replication process. So if we turn off the turn-off, then the cell goes, if we turn off the negative regulator, if we have a CHEK inhibitor, then the cell is going to go through the cell cycle. If it has damaged DNA, then it's going to be very susceptible to undergoing mitotic crisis. It's actually not apoptosis, it's called mitotic crisis. So interestingly, we found, which is consistent with what our hypothesis was, we found that the high-grade serous cancers were the most sensitive to the CHEK inhibitor in vitro. And they had a much lower IC50 in these 3 high-grade serous cancers compared to cell lines that were from patients with other histologies. So we have a clinical trial ongoing for CHEK inhibitor, and we have a BRCA mutation cohort, and we have a BRCA non-mutation cohort. And actually, our most promising results are in the non-BRCA mutation cohort. And this was highlighted by a functional genomics approach and not just a survey of genetic aberrations. This is a summary of the responses to date so far. Women with germline BRCA mutation were not as sensitive to the CHEK inhibitor or not as dependent on the CHEK pathway as compared to women with high-grade serous ovarian cancer without a BRCA mutation. And you can see here again, this is the response on the y-axis where 20% growth is considered progression, 30% shrinkage is considered response. So we have a lot of patients with stable disease in the interim category, and we have a bunch of patients with response, and the little pluses indicate that they are still on study with stable responses or stable disease. This is showing you that we've had a remarkable amount of time on study, and like I said, these women are still on study, and these are the ones with the responses. So this is a very exciting avenue for non-BRCA cancers. So I showed you the olaparib and carboplatin for the BRCA mutation cancers. The CHEK inhibitor seems to be very exciting for the non-BRCA high-grade serous cancers. Okay, so just to summarize, as I mentioned, ovarian cancer genomics. Ovarian cancer itself can be divided into epithelial, which is what I focused on most today. Non-epithelial cancers I did not focus on, but there are specific mutations for these types of cancers as well. And I focused mostly on the high-grade serous cancers, which have p53 mutations, BRCA1, BRCA2, and other pathway alterations. And now I would add in the CHEK1 functional dependence, because it's not actually an amplification or a mutation. So functional genomics back in 1981 was looking for transforming genes from whole chromosomes chopped up and put into fibroblasts. Now we have a functional approach looking at what we can knock down and what is actually important for the cell to grow. And if we put those together, we have a functional genomic screen. We identify a driver pathway, and then we perform a clinical trial as proof of concept to get the drugs into the clinic for patients. And with that, I will acknowledge all the people in the women's cancer team, translational scientists who worked on the CHEK in vitro work, and then my collaborators, of course. So I'll take any questions. Yes. That is a very, very interesting question. So people are definitely looking on that, whether the patient, certain mutations make you more susceptible to one or the other. So far, it's not clear. There might be other, methylations going on in the genome, or other, factors, epigenetic factors that might be influencing that as well. What we do see is that a lot of the women who have ovarian cancer have already had breast cancer, so it's not like it's really one or the other. So they might have had breast cancer that they found at an early stage and then had surgery to remove that in the chemotherapy. So they've been cured, or the breast cancer is in remission at the time that they develop the ovarian cancer. So a lot of people do have both. For some reason, the ovarian cancer tends to develop later in life compared to the breast cancer, so that's why it seems to go in that order. But these are all areas of active research. Okay. Ooh, I'm stuck. There we go. Okay.
So we have one class announcement, and that being you should have all received an email about the core facilities that you can sign up for. I lined up visiting the pathology core, small molecule robotics core, or genomics core. And then if you want to visit tumor boards, we have an arrangement with the medical oncology as well as the urologic oncology. So you're supposed to indicate your choices and send an email back to me by the end of the week. And then we'll be visiting the core facilities and the tumor boards, especially in October and November. Okay, so let's move on here. To our next talk. And so our next speaker is Stephanie Goff. She got her MD at the University of South Florida in Tampa. Subsequently, she did training in Steve Rosenberg's lab, and she did a surgical residency at Columbia and a surgical oncology fellowship at the Dana-Farber and Brigham and Women's Hospital in Massachusetts. And subsequently, she's come down to NCI. She's a member of the surgery branch. And if any of you saw the First in Human television show, it was on the Discovery Channel last month. She was one of the star staff clinicians treating a melanoma patient. Stephanie.
Can you guys hear me okay? All right, so slightly different than the talk you just had because I'm not going to talk about a lot of our own work in the surgery branch, but more to give you a feel for what's out there with immunotherapy in what we call the real world. So not inside the clinical center, but things that are going on outside. Make sure I do this right. So these are just the objectives for today's lecture. We're going to talk a little bit about the basics of immunotherapy. We're going to talk about the mechanism of action of checkpoint blockade. I am going to talk about our early clinical experience with the checkpoint blockade inhibitors here at the NCI, only because it's unique in sort of the development of this drug and how we discovered what immune-related adverse events were, defined them, and defined how to treat them. Then I'm going to talk a little bit about the poster child for checkpoint blockade, which is melanoma, and talk about Ipilimumab, Nivolumab, and Pembrolizumab, which you may know by the names if you've seen them on TV, Yervoy, Opdivo, or Keytruda. I'm then going to talk about checkpoint blockade and other cancers, and I have to update these slides almost every 3 months because there's something else getting approved for use in checkpoint blockade. And then just touch a little bit on the experimental questions that are happening in checkpoint blockade. So oncology, as you know, it requires a lot of different things to treat a patient with cancer. And if you had asked me 10 years ago, this would have been a Greek portico with just 3 columns. One of them being surgery, which is the oldest form of tackling cancer. We've actually seen old Egyptian papyrus with people doing an operation, so it dates back that far. Radiation came about in the early 1900s. Chemotherapy joined in the 40s and 50s and then really took off in the 70s. And then recently we've added immunotherapy to that. So we now have 4 different tools in our toolbox to try to help our patients with cancer, with immunotherapy being the most recent. The sort of targeted therapies, I think, sort of fall, depending on what their target is, in between chemotherapy and immunotherapy. So when we talk about immunotherapy, there's sort of 3 general ways that you can think about it. One is sort of a non-specific stimulation of immune reactions. So that's sort of trying to train the immune system that you already have in your body, or to stimulate the immune system that you already have. And that's by either stimulating effector cells, so turning them on, or inhibiting regulatory factors, so preventing them from getting turned off. And then the second way is to actively immunize, so train the cells while they're in your body. So cancer vaccines are attempting to do this. We've studied them in the past, and we're getting ready to start studying them again in a slightly different way. And then to passively transfer an activated immune system, so train the cells outside the body and then give them back something called adoptive immunotherapy, which is really what our specialty is at the surgery branch. But what I'm going to talk to you about today is checkpoint blockade. So the cells of the immune system, you are far closer to learning about these than I am, but primarily we're talking about T cells and what their effects on cancer can be. As many of you know, it does split into an innate immunity, or a very rapid response, which are these cells on the right side, left side, and then adaptive immunity, which is more of a memory response, but can be stronger. So when a T cell is born, it goes through the thymus. And it's in the thymus that it becomes, one of 3 things happen to it. It either doesn't get stimulated at all, and so it dies because it's not seeing anything in the body that it should recognize. Or it gets stimulated too much, meaning that there's something in the body that's everywhere and you don't want to attack it, so it dies from overactivation. Or it gets stimulated just enough. That's the Goldilocks. And then it will come out of the thymus as either a committed CD4 cell or a committed CD8 cell. And this builds within our body a repertoire of T cells. And I think I got the question last year, so I went back and looked this up, but we have about 4 × 10⁻¹�11 circulating T cells in the body of an adult human. Because of the way that our T cells train themselves, you can have anywhere from 10⁶ to 10⁷ different unique T cells in your body that recognize 10 to the 6th or 10 to the 7th different things. And we exploit that a little bit, and we'll get into that more. So when a T cell gets activated, it's sort of a 3-step process. The first is that very key to T cell understanding is that it has to recognize its antigen. It is a lock running around in search of a key. It's not searching for something that sits on the surface of a cell. It's searching for something that's come from within the cell and has been processed and put up onto the surface of the cell in something we call a MHC molecule. If you want to think about MHC molecules, they're sort of like the recycling bins of the body. As a protein gets degraded, a certain little piece of a protein will fit in the groove of an MHC molecule and then get put up onto the surface. And it's shown to all these passing T cells, passing APCs, hey, I'm here, do you want to come attack me? And if it's something that your body knows that it should attack, like a piece of a virus or something like that, it will attack it. And what we're trying to do is see if we can exploit the way that it attacks that. So signal 1 is what gives a T cell its specificity. That 10 to the 6th or 10 to the 7th different types of T cells that I talked about, that's the specificity or the antigen that T cell engages. Next is whether that T cell gets turned on or gets turned off, and that's dependent on signal 2. Now this is where the co-stimulatory molecules come into play. So at the same time that lock meets that key, there are other particles on both the target and the T cell that will then click together. And if it happens to be something that activates a green pathway, then the T cell gets turned on. If it's something that happens to activate a red pathway, then the T cell gets turned off. Checkpoint blockade that we're going to talk about is mostly a signal 2 phenomenon. The third signal, which is the one that most people haven't figured out yet an appropriate way to target with drugs, is because it's sort of the environment in which that T cell sees signal 1 and signal 2 depends on what kind of cell that turns into. Now in the lab, this is very important because we can, in the lab, generate either helper 1 cells, helper 2 cells, regulatory cells, or Th17 cells. We can't exactly manipulate the levels of cytokines in the body to create those just yet. We're trying. But it's very helpful in the lab when we're trying to learn what different subsets of T cells can do. And very few people have this pathway memorized, but it's there if you would like to look at it. Essentially, IL-2 and other pro-inflammatory genes are the end result of all of this activation. So what is the role of the signal 2 checkpoints? Why do we have them? I mean, the immune system wasn't built to conquer cancer, so how are we turning that around to make it go after these cancers that look so much like ourselves? So the immune checkpoints promote a self-tolerance. It's to prevent us from attacking things. So the initial response to antigen occurs primarily in the secondary lymphoid organs. So those are your lymph nodes, your tonsils, your spleen. Peyer's patches, if you want to think of them, are just little lymph node patches that live in the gut, in your small intestine. Mucosa-associated lymphoid tissue. It prevents our body from attacking the proteins that it sees, or else we'd all walk around with autoimmune disease. So what these immune checkpoints do when they're functioning normally is to limit collateral damage. So if you get a splinter and it has a bacteria on it and your body knows to go after it, you want your body to stop going after it after it's killed the bacteria, because if that inflammation keeps going, then you have a non-healing wound. So what these immune checkpoints do from an evolutionary standpoint is to shut down the immune system before it gets out of control. But for cancer immunotherapy, these checkpoints create opportunities to break that tolerance to self-antigen. CTLA-4 is the first of these that we knew about, discovered by Jim Allison, while I believe he was at University of California, San Francisco. It is a negative regulator of cell activation. So both naive, so T cells that have never seen their particular antigen in the wild, and memory T cells express something called CD28. When a T cell sees its antigen, if it clicks onto CD28, it will turn on. When it turns on, part of that negative feedback loop is that CTLA-4 will then become transported to the surface. So you've got a resting cell that doesn't have any CTLA-4. When it gets engaged, these vesicles full of CTLA-4 will come to the surface. Then this CTLA-4 will bind and shut this T cell down. It becomes a negative regulator. So it's a negative feedback loop to dampen the downstream processing. So naive and memory T cells normally don't have this. They have to wait till they get engaged. On the other hand, Treg cells or T regulatory cells, those cells that we think play a role in helping prevent our bodies from sort of destroying themselves, always have CTLA-4 on the surface. So they're always capable of being shut down. PD-1 is a second area of checkpoint, and this is sort of in conjunction with the work of CTLA-4. Different than CTLA-4, which is at the initial antigen presentation, this is when the T cell gets out to the tissue. So this is more of the splinter type phenomenon. Once the T cell engages the tissue, the fact that it starts to kill and release interferon gamma and things of that nature, the inflammation that's generated around this tissue will then make it express PD-L1 or PD-L2. What that does then is give the PD-1 on the T cell the opportunity to get turned off. So again, it's inflammation present in the tissue that's promoting the upregulation of the ligands, and this limits the collateral damage. So again, on a normal day-to-day basis, when you're not talking about cancer, having these checkpoints around are very important to prevent autoimmune disease. Now, what we know about certain forms of cancer is that the cancer cell itself sometimes will have epigenetic modulation or increased upregulation of the genetic pathways so that PD-L1 becomes overexpressed on the cancer cell. It doesn't wait for inflammation to show up, it's just constitutively present. When that happens, these T cells can get shut down. Secondarily, when we learn how to go after one, when we've got a successful T cell engaging a tumor cell, that's when this upregulation happens. So that's the negative feedback loop. So most of the cancers in which we use these to target take advantage of this situation, but there is one cancer in particular that takes advantage of this, and we'll talk about that a little bit later. But when we find a successful immunotherapy, our success actually becomes a hindrance because the release of interferon gamma, which signals T cell killing, also stops T cell killing. So when we started to think about checkpoint blockade after Dr. Allison found that CTLA-4 could work and it could work, blocking it could work in mice, where would we start? So what we started with were tumors that were known to respond to other immunotherapies. So melanoma, as many of you may know, in the metastatic setting, there are an estimated 10,000 deaths per year in the United States. Metastatic disease, once it spreads, has a 5-year survival that's pretty dismal. Only about 16% of those patients survive. We do have a drug called interleukin-2, which is one of those drugs I talked about in the beginning, which just turns on all of the cells that you have in your body. What we found through careful use of that is that it could actually cure people. So cure isn't a word you hear us use a lot. We wait until patients are 10, 15, 20 years out before we use the word cure. So when we treated close to, I'd have to go back to this paper to be certain, but I think it's close to 600 patients with IL-2. 4% of them had their disease go away, never come back, greater than 10 years. We consider that a cure. So we've taken these patients that have a 16% 5-year survival, and they're alive 10 years with no disease. So immunotherapies can work in melanoma. We knew that going into this. Kidney cancer or renal cell cancer, slightly more prevalent than melanoma, with an estimated 14,000 deaths per year in the US. Slightly worse prognosis, with only 12% surviving 5 years once they develop metastatic disease. but even more responsive to immunotherapies with a durable cure, again, more than 10 years with no evidence of disease in 7% of patients. So this was 2001. We got started with this particular clinical trial. And what we started was with this drug against, the A is just anti, so an antibody against CTLA-4, which was called Ipilimumab. So as you're hearing more about monoclonal antibodies, just to keep in the back of your head, MAB means monoclonal antibody. So if you see a drug name that ends in MAB, it's a monoclonal antibody. The next two letters next to that will give you an idea of where it came from. MU is murine. This is an antibody that was raised in a mouse. LI is the target. Either using I or LI means that you're targeting a portion of the immune system. And then the IPI is proprietary to whoever invents the drug. So this naming pattern lasted just up until recently, when finally the WHO abandoned it. But for most drugs that are out active today, you can actually look it up on Wikipedia. It will tell you what all the different chunks of the word mean. But the most important to remember without knowing the others is that MAB is monoclonal antibody. So this was a phase 1 trial, and I don't know if you've gone over what the phases are, but essentially that means it's the first time it was tried in humans. And traditionally, what you're testing for is a safe dose. So we had this monoclonal antibody. It was given at 3 milligrams per kilogram. We also injected a peptide that we thought could help boost this like a vaccine. We enrolled 14 patients. We had 2 complete responders and 1 partial response. But what happened was that accrual was stopped for toxicity. So in any sort of phase 1 drug trial, you have a particular dose. And if you run into problems or toxicities or side effects, you have to stop and evaluate whether that dose is correct, whether you need to come down on that dose. And with traditional drugs, what that means usually is that you need to come down on the dose of the drug. So these are some of the responses that we saw. They were real. This is a solid tumor sitting in a patient's lung. I don't know how familiar you are with CAT scans, but if you want to think about the fact that the patient is lying on the flat with his head behind the screen and feet out towards you. So this is the right side of the patient. This is the left side of the patient. This is the patient's back. This is the patient's front. What we're looking at here are lungs. The black areas are the lungs. This is the heart in the center. And what you're looking at here, this blob, shouldn't be there. That's metastatic melanoma. And what you look at here on this other side is that blob is gone. Okay? What we're looking at in the lower panels was we found this is skin. And you can see there's an infiltration of these little dark purple cellular structures. Those are T cells coming to the surface of the skin. And all of this normal skin structure underneath has been destroyed by the immune system. That was a patient who had dermatitis. This is colon, and you're seeing normal colonic glands, but then you're seeing, again, these small purple nucleated cells. Those are lymphocytes. Those shouldn't normally be in the middle of the colon. This is just yet another example of that with a different sort of stain. And this is normal liver that's been infiltrated by T cells as well. So the FDA told us to stop till we figured it out. And then because we were treating this like any other drug, we actually dropped the dose. So we did a dose reduction going from 3 milligrams to 1 milligram. But what we were finding was that if patients developed these toxicities, so grade 3 means that it was fairly significant and we had to treat it with something other than just creams or Benadryl. Usually this meant we had to treat it with steroids. If someone developed a greater than grade 3 adverse event, they were more likely to have their tumor go away. So this was a first. So in the development of regular drugs, non-immunotherapy drugs, The more toxicity you got never corresponded with more response. It just meant the patients got sicker. In this, what it was actually showing us was that if we were activating the immune system, we were activating it against all these normal things, but we were also activating against the tumor. So then it became a risk-benefit payoff for these patients. How much are they willing to tolerate for the chance of having their tumor go away? So this was in melanoma. It was 36 to 5%. And in renal cell cancer, 29%, none of the patients that didn't have a grade 3 adverse event responded. So we felt we need to push it a little more. So this created a whole paradigm change at the FDA, one that has then led to how we are able to evaluate checkpoint blockade today, in that they allowed us to do what they called an intra-patient dose escalation. So they let us go up on the dose of the drug, but only in each patient. So we would start with 3 milligrams in a patient. If they tolerated it okay, then we were worried that we weren't getting the effect we wanted. So then the next dose we would go up to 5, or the next 2 doses we would go up to 5. And if they still weren't seeing anything, we worried that we weren't triggering the immune system enough, and we actually went up to 9. Because we wanted to see if this relationship between adverse events and response was true. So we did a formal phase 2 dose escalation, and again demonstrated this association of response with immune-related adverse events. So again, this was in patients with melanoma. We had 50 patients develop a grade 3/4 immune-related adverse event. 28% of them had an objective response. We had 36 patients develop a lower grade, meaning usually didn't require much treatment. 22% of them responded. We only had 1 patient respond out of the 53 that did not have any immune-related adverse events. Not all adverse events are equal. A bad dermatitis that requires a skin cream is one thing, but we had some bad enterocolitis, both in our patients with melanoma and renal cell cancer. So what does that enterocolitis look like? It can be a complete destruction of the crypts of the colon that could lead to requiring an operation for perforation. So this was the most serious. When we could catch it early and give steroids, we were able to stop that from progressing to needing an operation. But more importantly, giving steroids or shutting down the immune system, which is what steroids are generally thought to do, didn't shorten these patients' responses. So giving steroids didn't hurt what we were doing, but it did protect them a little bit. So we were able to come up with a strategy for treating the toxicity without losing the tumor response. So it sort of concluded the work we did at the NCI, because our job here is to really sort of test things and then get them out into the world and let companies figure out how to make them commercially successful. So we sort of stepped back from the development of Ipilimumab. But what we did during the phase 1 and 2 is to develop algorithms for management of these immune-related adverse events. And over time, we were able to demonstrate that they were durable. So one of the key things with checkpoint blockade, and I urge you to look at this as we're going through more trials as I finish up my lecture. Traditionally, chemotherapy, this is a graph of overall survival, so a patient drops off of this if they die of their disease, is that most trials of chemotherapy, you'll have patients that don't do very well in the beginning, so this will be very steep, and then they'll sort of split like these red and blue lines do, but inevitably they usually come back together and they come back down to 0 because none of them are curative. So the patients all at some point end up dying. What we like to talk about in immunotherapy is the tail of the curve. That's one of the unique features of immunotherapy and checkpoint blockade is this tail of the curve. So you've got patients here, this was a trial where we used Ipilimumab in combination with IL-2, and here at 60 months is 5 years, you've got 20% of them still alive. Even in this initial trial of Ipilimumab, the very first one that we closed because of toxicity, it's only 10%, but 10% of them are still alive now almost 10 years. And in the second one, when we were escalating the doses, you see that it got a little better survival when we escalated the doses. And again, there's a tail of the curve. So anywhere from 10 to 20% of these patients are still alive 6, 7, 8 years later. So that's the big difference between standard chemotherapy and immunochemotherapy or immunotherapy. So this is just a schematic of what I've been talking about. So either the tumor or the antigen presenting cell will present an antigen to a T cell. CD28 is a positive, CTLA4 is a negative, PD1 is a negative. Anti-PD1, which we're about to get into, is Nivolumab and Pembrolizumab, block here. Ipilimumab blocks here. So in melanoma, again, I just urge you again to look at the tails of the curve. See how these are going out. So there was only an 11% response rate. This is after we finished our trials here and sort of passed it off to the company, who then got a huge national trial going with a consortium under Dr. Hody at the Dana-Farber, who then published his findings. An overall response rate of 7%, which doesn't sound so great. 38 out of 540 with a complete response in just 3 patients. And what you see here is progression-free survival. These patients, their disease came back. But again, the tail of the curve, even when it came back, they're still living longer. So there's something about activating their immune surveillance that helps keep them at bay. And then this was approved for metastatic melanoma in 2011. It was the first new drug for metastatic melanoma since the 70s. Take that back. IL-2 was in 98. It was the first one since then. They've since updated the survival on this. That tail of the curve has held now that we're out 7, 8, 9 years. So durable, though for a small number of patients. Tremelimumab, again, monoclonal antibody raised in a mouse against an immune target, but made by a different company. Didn't do as well. And the reason why is that it didn't separate the survival curves enough. So this is actually one of those drugs that is now used sort of to test the concept in mice, but it's really not gone any further in human use, although it's being used at the NCI by a few groups in combination with radiation therapy and other combination trials. But it didn't gain any traction as a standalone drug. Nivolumab, monoclonal antibody. They cheat with the MU and the U. U is also mouse in this instance, and L is the immune, and then the Nivo is proprietary. They did a phase 1 dose escalation. They didn't know which drugs these are, which cancers this was going to work in. So they opened it up to a bunch of things. So not only things that had seen benefit with ipilimumab, so melanoma and kidney cancer, they threw in things that were common. So lung cancer, hormone-resistant prostate cancer, and colorectal cancer. The type of plot that you're seeing here is called a spider plot, sort of for obvious reasons, in that every patient starts at 0, and then this tracks the % change in their tumors. So what you want is to go down. What you really want is to go down and get to 0. What you're okay with is to go down and stay stable or stay stable. What you don't want is to go up. And what you don't want is to go down and then go up. But you can see here from the spider plots, most of these are going down. Interestingly, there's this one delayed that didn't go down until almost 2 years of therapy. But what they saw was that, yes, it was active in melanoma. It was active in lung cancer. It was active in renal cancer. No reactivity in colon or prostate. And again, we're looking at CAT scans again, because the novel finding here was that it could work for lung cancer. And so you want to demonstrate that that's actually real. And so you're looking at this tumor here, this tumor here. It actually got bigger, which we think is from a swelling of the immune cells sort of rushing to the area, and then went away. Again, bigger, went away. They weren't seeing as many toxicities with this drug as we did with ipilimumab, and we think that's basically because this works more out toward the periphery when the the immune cell engages the target rather than in the center when the immune cell is learning how to be an immune cell. This is another kind of plot that you'll see when you look through clinical trials. This is called a waterfall plot, and this just lines up every patient in the study. And instead of seeing a spider plot with all those things, it will take the deepest point of the spider plot for each patient. So all the patients over here, these 3, 4, had all of their tumors go away. They go all the way down to -100. Everything at this inflection point, their tumors grew on therapy. Everything they went down. This dotted line is at 30%, which is just sort of the recognized international standard for declaring a response. And so you can see these patients are all considered responders. The little asterisks, which I don't know if you can see them, they're rather small, but that means that that is ongoing, so that those patients haven't progressed. This was a randomized clinical trial, or RCT. So this was nivolumab versus chemotherapy of choice, although I will tell you there aren't very many good chemotherapies for melanoma. Dacarbazine was approved in the 70s. It can have a very short response in about 30% of patients, but it always comes back in about 4 months. These patients all had to have, because ipilimumab had been approved at this time, they had to have been refractory to ipilimumab, meaning that they either had a response and it grew back, or it never worked in the first place. A response rate of 32% with 4 complete responders. So that definitely showed promise. And it was approved for refractory melanoma in December of 2014. Well, now they can use it for patients who have already gone through ipilimumab. And if I mess this up, I want you to correct me. I always say ipilimumab failed the patient, not the patient failed the ipilimumab. So it's now approved for those patients. So what they wanted to see was, given all the toxicities that come with ipilimumab, can they get it approved without having to see ipilimumab first? So then they needed to do a randomized controlled trial. This is nivolumab versus dacarbazine in the untreated setting. So outside the world of checkpoint blockade, there were drugs being approved for BRAF mutations, which is completely separate from checkpoint blockade, but because patients who were BRAF mutated could have another approved drug, they were excluded from this trial. So you had to be wild type BRAF. What they found, you won't see curves like this in almost any cancer trial today. The differences are just too great. These are patients who got nivolumab. These are patients who did not. The curves split nicely, and the nivolumab is starting to have a tail. Now, granted, this is very early. This is only a year. But with this sort of separation, you would expect that the nivolumab is better. And in fact, they updated the survival for the tail of the curve. And I forgot to put in here, but it did get approved as first treatment for melanoma. This is a second drug, excuse me, pembrolizumab, again, MAB. Now, ZU means that it is a humanized antibody, which means that, yes, it's raised in a mouse, but that mouse has been genetically engineered to have human immune system. So a humanized antibody against a portion of the immune system. Similar trial like we've talked about before, it's looking for a dose, and it had a 26% objective response rate, and 12% had grade 3/4 AEs. And it was approved for refractory melanoma in September of 2014. This was just testing the dose. We don't need to go through that necessarily. And then they wanted to go as first-line therapy. So they tested it in actually what many feel was a better way of testing. The last randomized controlled trial tested nivolumab versus dacarbazine. Nobody uses dacarbazine. So this one tested pembrolizumab versus ipilimumab, which is actually what patients were getting. They tested 2 different dose strategies, Q2 week versus Q3 week. So 1 infusion every 2 weeks versus 1 infusion every 3 weeks, and they randomized 1 to 1 to 1. And you can see here that in both arms, pembrolizumab outperformed ipilimumab. But what's more astonishing here, There were 31 people who had complete responses and are potentially cured with this drug. Again, survival curve splitting in ways that we usually don't see. This is progression-free survival. This is overall survival. Now, what's important to remember is that all of these drugs are checkpoint blockades, so they all are going to have this tail of the curve. What you saw, though, is that, remember, pembrolizumab had a higher response rate. It had lower toxicity problems. So it appeared that pembrolizumab would be a gentler way to achieve the sort of immune responses that we were seeing with ipilimumab. Again, they did update their survival. This is their overall survival here, and you can see the tail of the curve now at 2 years, still relatively impressive. Over 40% of all the patients that got ipilimumab are still alive, and close to 60% of all the patients that got pembrolizumab are still alive. So when we talk about checkpoint modulation, we're talking about an interactive series of a bunch of different receptors and ligands. This being the antigen presenting cell or the tumor cell, this being the T cell. And what we've talked about is a way to block this, which is ipilimumab, CTLA-4, and a way to block this, which is PD-1. What if we blocked them both? Could it improve response or survival? So this took patients who were previously untreated. It was a phase 3, so randomized controlled trial, and it did 3 arms again. Nivolumab, so anti-PD-1, nivolumab plus ipilimumab, anti-PD-1 and anti-CTLA-4, and ipilimumab, just anti-CTLA-4 alone. Their thought was that you needed to have PD-L1 on the tumor or the antigen presenting cell in order to get a response to PD-1. And so they tested that hypothesis by checking PD-L1 positivity. What they found was that 44% of patients had an objective response to Nivo, but close to 60% of patients had an objective response to the combination, whereas only 20% had a response to ipilimumab alone. But almost more than half had a serious grade 3/4 adverse event in the combination. So there's something about letting loose the immune system in 2 different ways that makes it much more powerful against the cancer, but also makes it much more powerful against normal tissues. When you look at patients who were PD-L1 positive versus PD-L1 negative, you can see that there was perhaps a slight difference, but not really as big a unique difference as the investigators thought there might be. The hypothesis was that if you didn't have PD-L1, then the addition of nivolumab shouldn't help, but it turns out it did. Still with an overall response rate of 50% versus 30%. So they updated this. Their overall survival is still not updated. This was at ASCO a year and a half ago, even at this past year's ASCO, the American Society of Clinical Oncology, they still had not yet updated it. But you can see these tails of the curve, which we're getting more and more used to seeing in immunotherapy. And the combination was approved in January 2016. So why melanoma? So at the same time all of this was happening, a lot of the work that Dr. Nunziata was just talking to you about genomics and proteomics and epigenetics was starting to mature. And the TCGA finally published their database on all the human tumors that they had. And what they wanted to demonstrate was that different tumors had different rates of mutation, meaning that if you compared a patient's tumor to a patient's DNA, there would be certain portions that had what we call non-synonymous mutations, meaning just a single change in a residue that led to a different amino acid. Excuse me. And what we learned from that is that melanoma tends to be one of the more highly mutated tumors in the genome. And a lot of that is predicted to be because of UV damage. So UV causes a DNA breakage. When that DNA has to repair itself, it runs the risk of creating a mutation. And it turned out to be very high. What we also found out was that lung cancer, because of the toxins associated with smoking, also has a high mutation rate. Bladder, which is not something you would normally think of, but every toxin that's in your body ends up getting filtered through your kidneys, not every, most. And so it ends up in your urine and sits in your bladder. So any chemoirritant, any toxin that's there could create a mutagenic environment in the bladder. This is a wonderful paper for those of you interested in the genomics of mutations that came out in Nature in 2013. So is it because melanoma is mutated that it responds to checkpoint blockade? So we looked at other highly mutated tumors. So non-small cell lung cancer, the first thing you notice is that it's an order of magnitude or two more deaths per year than melanoma. So close to 160,000 deaths per year in the US. It has an even worse prognosis for metastatic disease with only a 2% 5-year survival. And there is a correlation between smoking and the number of mutations that are present. Bladder cancer, 16,000 deaths per year and highly lethal once metastatic. And then this unique blend of tumors with mismatch repair deficiency. So mismatch repair proteins help our body's DNA repair things after it breaks. You can have a germline mutation, which is called clinically Lynch syndrome, and you can have a sporadic mutation in one of these 4 proteins. What we found when we sequence tumors with a mismatch repair deficiency is they have on the order of 15 to 20,000 mutations per genome, whereas a melanoma will have on average about 200 mutations per genome. Melanomas being the most mutated of all the known human cancers, and still it's 2 orders of magnitude less than what these germline mismatch repair tumors will have. So nivolumab was tested in non-small cell lung cancer. Again, because there are a lot of other approved treatments, they had to be refractory to at least 2. They were enrolled on a phase 2 study called Checkmate 63. I give all these references and numbers in case you want to do some further reading on your own. 3 milligrams per kilogram every 2 weeks. They treated 117 patients. Again, this is a waterfall plot. In this particular one, they've told us whether patients are alive or dead. And you can see the responders are all still alive. And even some of the non-responders are still alive. But for the most part, if you progressed through this, you did not survive your lung cancer. They did a randomized controlled trial in one particular kind of non-small cell lung cancer, which is squamous. So they tried nivolumab versus a standard chemotherapy called docetaxel. These patients had already been through one platinum-based regimen to begin with. Again, you see the split in the overall survival. Nivolumab with a 20% response rate, docetaxel with a 9% response rate. FDA approved nivolumab for refractory squamous non-small cell in March of 2015. What about the non-squamous or the adenocarcinomas? They did the same trial, nivolumab versus docetaxel, saw slightly different results, but what was clear was that the hazard ratio for death was on the side of the nivolumab. And so this got approved for refractory non-squamous in October 2015. Well, if nivolumab works, pembrolizumab should work. It's just a matter of whether that drug company can investigate it as thoroughly as the other one can. And so they tested 495 patients within a larger trial. And what they found was that smokers had a higher response rate. And why do you think that might be? Because the smokers had higher numbers of mutations in their tumor. So it's kind of like the concept of having a lottery ticket. If a mutation is a lottery ticket, if you've got more of them, you've got more chances of winning the raffle than if you've got less. And this was approved for advanced non-small cell lung cancer in October 2015. What you'll see with nivolumab and pembrolizumab is they tend to go back and forth, with one getting the approval first and then the other one following a few months later. It's just a matter of who gets it first. But now there's a new agent that we haven't discussed before, which is anti-PD-L1. So this doesn't block the PD-1 side of it, it blocks the PD-L1 side of it. And because this was sort of an area that not a lot of people were looking at, they jumped into urothelial bladder cancer. So why weren't a lot of people looking at it? Well, lung cancer, melanoma, all these others tend to be diseases of the young and the middle-aged. There's a lot of them. They have a lot of advocates. There are a lot of pushing for new drugs. Urothelial bladder cancer, the median age is in the 70s. And so it's just sort of an unmet need until some companies took an interest and decided to test their drug in that population, knowing that bladder cancer was very highly mutated. So atezolizumab, again, the MAB, ZU because it's humanized, LI because it's against part of the immune system. The tumors, they measured their PD-L1 just to see where they lie, and they had 65 patients that they could treat. And what they saw was that if you had greater than 5% PD-L1 on your tumor, you had a 43% response rate, versus if you did not, you had a lower response rate. So it may be that the presence of PD-L1 does help distinguish those responders from non-responders. So in this waterfall plot, the different colors that you're seeing just demonstrate that it's not an absolute. So the ones in green, the light green being what they call an immune histochemistry, an immune score of 2, and the light green being immune score of 3, that is a combination of how much PD-L1 was seen on biopsy. So it's not perfect. Yes, there are a lot of green and dark green patients way over here in the good responder bit, but there's a couple of them over here that did not respond either. Vice versa, the orange and the red, most of them did not respond, but there were a couple that had responses. So it's not perfect, but it does seem to delineate a little bit. And this was just a different trial looking at overall survival. This wasn't randomized, which is why there's only one graph. But what you can see, to have a 1-year survival rate of over 20% is almost unprecedented for urothelial bladder cancer. And this got approved in May of 2016. So it lost its number, MPDL 3280A, and got a name, which I think is Tecentriq. So go back to some of the old standbys. Can we get pembrolizumab to work in urothelial cancer? Well, they tried it, and they did see an objective response rate of 29%. You can see from how well you can actually see the steps here that there aren't a lot of patients in this trial. But even with that, at a year, you've got a 30% patients still alive. And nivolumab as well was part of a bigger study. Again, with a median survival of close to 10 months and 45% of patients alive at a year. So what about those other highly mutated tumors? We talked about lung cancer, we talked about urothelial cancer. How about the mismatch repair deficient cancers? So it builds on this hypothesis of neoantigens developing from somatic mutations. And this was a phase 2 study, and they had 3 cohorts. So colorectal cancer that does not have a problem with mismatch repair genes was the first. Colorectal cancer that does have a problem with mismatch repair genes. And then other cancers that have mismatch repair problems. So in the patients or in patient cohorts, patient families that carry Lynch syndrome, they'll develop endometrial cancers and other cancers, and they wanted to test that as well. How they have color-coded this, I'm showing you both a spider plot and a waterfall plot, but those that are mismatch repair proficient, meaning they do not have a problem with repairing their DNA, less mutations, are the red. The blue and the black are those that have a problem repairing their DNA, and you can see almost all the blue and the black go down, almost all the red go up, almost all the blue and the black are over here, and almost all the red are over here. So it seemed to be a pretty clear signal that if you had problems repairing your DNA and you had a number of mutations, that you were more likely to respond to this therapy. Excuse me. They actually showed some immunohistochemistry of that. So this is a patient who is deficient. This is a patient who is proficient. So this patient can repair DNA without issue. This one cannot. This is normal tissue. This is tumor. What you can see is there's high levels of PD-L1 on the tumor in a patient who is MMR deficient, where there's very little in the proficient. There are a number of CD8 T cells where there are few here. This is at the interface of the tumor and the normal. If you go deeper into the tumor to look for tumor-infiltrating lymphocytes, these are in two different subjects, but this one is MMR deficient. There's a huge influx of tumor-infiltrating lymphocytes. So something about the fact that these cells have damaged DNA, they have things that T cells can see. The T cells get drawn to the center of the tumor, whereas that's not happening with increased frequency in the proficient cancers. And this was just approved within the last few months. It's the first time the FDA has approved a drug not based on what kind of cancer it is, but based on the genetic makeup of that cancer. It was a big first. So it didn't matter what kind of tumor it was. It could be a breast cancer, an ovarian cancer, a colon cancer, a bile duct cancer. If it is mismatch repair deficient, it's now approved to get pembrolizumab. And it's the first time that's ever happened. Of course, one follows the other. Nivolumab did the same study. There is a second name for these tumors, which is microsatellite instability, those patients with microsatellite instability, or MSI-High. And again, they saw a high objective response rate, and you can see all those spider plots going down. And so Nivolumab attained approval for the same indication just last month or 2 months ago. So we know that it can work in highly mutated tumors. What about other tumors? So we know kidney cancer responds to immunotherapy. Remember earlier in the talk I said there were some tumors that would express their own amounts of PD-L1 just constitutively because that gene program got turned on. Hodgkin's lymphoma is one of those. So the Reed-Sternberg cell, which is the hallmark of Hodgkin's lymphoma, activates the gene signature to turn on PD-L1. Head and neck squamous cell cancers have a number of mutations. And Merkel cell carcinoma, because they are derived from the infection of a polyoma virus and UV damage, also have a high number of mutations. So I'll go through these quickly so we have a few minutes for questions. But they tested in kidney cancer checkpoint versus everolimus, which is an mTOR inhibitor, and found that there was a 25% response rate. You can see the survival curves split. Even though it's not a huge amount of split, it is enough to get it approved for kidney cancer. But what I will say is the bar is fairly low for kidney cancer. There are now, I believe, 9 different approved medications for kidney cancer. Most of them are of the small molecule TKI inhibitors that you will have heard of, like sunitinib and others that are NIBs. Astoundingly, in Hodgkin's lymphoma, so these tumors turn on PD-L1. I think the graph sort of speaks for itself. Almost all of the patients are falling to the right of the inflection point. 66% objective response, 7 of which are in complete remission from their lymphoma. So this was approved in May of 2016. It's very rare to see a plot like this. Of course, one follows the other. Nivolumab got the indication, so pembrolizumab came in as well. Again, you can see the majority of the patients. Now, they have many more patients in this trial, but still the majority of them, 69%, fall to the right of the inflection point. And they also got the approval for refractory, chemo-refractory Hodgkin's. In head and neck squamous cell cancer, again, you can see overall survival there with a response rate of 18%. Now, this is a very small study, but in this day and age, you don't really need large studies to get approval. So this got FDA approval in August 2016. Avelumab is an anti-PD-L1 drug, and you can see the spider plot is very colorful, but it's split sort of half and half. But it was able to show an objective response in 32% in a cancer that did not have any good options before this. In fact, Merkel cell carcinoma is one of the more unique cancers because we just figured out what caused it within the last 10 years with the infection of the polyomavirus. And this has been approved this year in March. So the PD-1, PD-L1 pathway, I've talked to you about a lot of different drugs that are there. If you go to this review and seminars of oncology, it will give you most of these numbers. I've updated a few. These have all been approved by the FDA. Bladder cancer has at this date 5 different PD-1, PD-L1 drugs that have been approved for its use in the metastatic setting. Breast cancer, which is the field that I'm trying to use immunotherapy in, has a little bit more of a mixed response. So if you have PD-L1 positive tumors and you are a triple negative patient with triple negative breast cancer, there's a chance that you can respond to therapy with about an 18% response rate. What they have shown recently is in first-line therapy of patients who are triple negative and PD-L1 positive, so patients who have not gotten any other chemo, for which there are multiple for breast cancer, there's a 23% response rate. But again, I caution you, this is a very small study when you think about how many women have breast cancer in this country. Gastric cancer, I didn't get a chance to update this slide because it just got approval for gastric cancer Friday of last week. I'm sure Nivolumab will be following shortly. So at ASCO the last 2 years, as you might imagine, people are trying to use these drugs in almost every disease that you can think of. The only one that hasn't really shown any signal at all is uterine leiomyosarcoma, but that's a relatively rare cancer type. But you can see there's a little bit of a signal in almost everything, and that probably has to do with the fact that the body doesn't care what kind of cancer it is, it cares if it has a mutation. So the concept is that if a cancer mutates just enough, your body no longer thinks of it as you. It thinks of it as foreign, and that's why it engages the immune system. And so it's sort of indiscriminate as to where the tumor comes from. It's more whether it has mutations. So these are all the different targets that we and other translational scientists are trying to go after. By blocking some of these in combination, will we be able to get even better responses to even more types of tumors? And that's what I got. Do you guys have any questions about anything?
So at the end of the curve, do you stop injecting the Nivolumab?
So that's something that's currently hotly debated. You can imagine pharma is on the side of giving the drug until patients progress. What is given now is that patients will get up to 2 years of therapy. When it's Ipilimumab, because of the rate of toxicities increases, they actually stop Ipilimumab after only 4 doses. So that's only 12 weeks of therapy. But the Pembrolizumab can go out to 2 years before it's stopped. Mm-hmm. Mm-hmm. Mm-hmm.
Have people been looking at these other drugs for people who might not have high mutational rates? Could you look at a sensitized patients to drugs using radiation to induce additional mutations?
So she's asking if we can use radiation to sensitize patients to perhaps respond better to some of these checkpoint blockade drugs. And what I will tell you is that there have been a few instances. In particular, there's a radiation oncologist who's now up in New York who treated a man with gastric cancer. He happened to have a very painful lesion that needed radiation. So she radiated that for purely palliative reasons, not research. But then the man was desperate and managed to talk the cancer center into giving him Ipilimumab. And despite it being something in which we knew ipilimumab didn't work, gastric cancer, and the combination of whatever happened to that man, it's anecdotal, but his disease is gone 4 years later. So whether it's the radiation sensitized something, whether it was an unusual combination of his immune system, I know that tremelimumab in particular is being used with radiation of a single liver lesion by one of the investigators at the clinical center now. So people are trying to use it to release antigen through radiation killing. All right. Thank you, guys.